Starting off with Jan Behrens, professor of physiology at the University of Freiburg. That's the German Freiburg in Breisgau. We will see later why that's important. And yeah, we have an add-on, the Opto kit for the Orbit Mini device, which can, through translucent recording chips, correlate optical and electrical readout, which is a super hot topic, and Jan Behrens will tell us all about it.
Yeah, okay. So I'm going to tell you about simultaneous fluorescence and voltage clamp measurements on freestanding membranes. And by way of introduction, I mean, pairing electrophysiology with fluorescence is an old thing in a certain way. I mean, it's been fashionable since some time because, of course, what you do is you want to know how ion channels move in the membrane, how they change their conformation to do what they have to do. Open, for instance, in response to a voltage change in the membrane. And here, this is all work from Isakov's lab, where they have labeled certain residues in a voltage-dependent channel, and they've seen that they can see fluorescence changes. But of course, these were fluorescence changes from very, very many channels simultaneously in a synapse oocyte. Similar stuff was done by Pancho Besania, for instance, in this paper here, where they had linked an Oregon green dye to the 3,4 linker in potassium channel, and they could actually show that they had a correlation between the opening of the channel, which is not shown here, and the change in fluorescence as the dye molecule was exposed to a different environment. Again, very, very many channels, millions probably of channels in a xenopus oocyte membrane. A little later, Richard Blunk in Pancho's lab did something involving COS cells, so small cells. That was, you could call that already something like whole cell voltage clamp fluorometry rather than the 2-electrode needle clamp. And some people also tried something similar in patches, but this was actually involving labeled agonists rather than labeled channels themselves. So even before those developments, there had been the dream, sort of the idea that you could maybe do this on a single channel level. So and Niels knows exactly what I'm talking about because he was also involved in some of the stuff that I'm going to show a little bit later. So the idea was you could maybe look at a membrane patch with a high resolution objective, something like this. It doesn't really look very high resolution in this thing here. And then you could directly correlate some fluorescence signal with the opening and closing of a channel. And of course, that would be wonderful. So these people here, McDonald and Ray, proposed this in 1995. In 1999, Shimon Weiss had a review article in Science and proposed a single molecule FRET experiment involving opening and closing of a channel gate and recording simultaneously the current. And of course, that was all kind of fantasy, So, but it was a fantasy that was well founded in the aim of directly correlating structural and functional changes, because the approach would be the single channel electrophysiology plus single molecule fluorescence, for instance, single pair FRET. And the reason is that only single protein measurements are capable of the time resolutions in ensembles will always smear out, So you will not be able to see, for instance, these changes in conformation that underlie the transition between different closed states of the channel before they actually go into the open state and correlate that with the functional signal, the ion channel. And here's a new notable from Paul Selving, who actually said that the holy grail of ion channel studies is to produce an atomic scale movie of an ion channel at work, simultaneously observing conformational and electrical changes as ions flow through the protein. And that's still the holy grail, so to speak. And this was actually written as a News and Views or New and Notable article about some work that Niels and I were fortunate enough to be able to contribute a little bit to. And that was actually the first experiment where some sort of correlation of that sort of thing was actually possible. This was together with Gerhard Schütz and are led by Gerhard Schütz and Andrew Woolley. And so the idea was, if you had a gramidine peptide, you know, this gramidine channel that occurs when two of these peptides come together in the membrane, and they form a channel that conducts cations, and you have one of them labeled with a fluorophore and the other with a donor fluorophore and the other with an acceptor fluorophore. And maybe you can also make it so that you can distinguish homomeric associations from heteromeric ones, meaning that you can distinguish whether one is donor and acceptor or donor and donor, which is not shown here, by modifying the donor labeled gramidine chemically so that it has a lower conductance. So then you can have three types of channels. You can have the high conductance dimer, the low conductance dimer, and you can have the heterodimer. And that tells you what your combination is. Yeah? And so this heterodimer conductance is the one for which you would expect to see a Fred signal. And in fact, there was some sort of correlation visible. Yeah? And there's actually a very fair appraisal of this in a... Sorry, is that... Oh, no, yeah. Let's go here. This is the wrong sequence. In this article here by Cargill and Baron Chanda, where they wrote that this was actually the first report of simultaneous observation of single molecule fluorescence and so forth. And they showed that association of monomers of Psi3 and Psi5 labeled gramidine, so this is the donor, this is the acceptor, shown by Fred, correlated with single channel opening in some recordings, but not in others. I think that's pretty fair. And also they say, well, there have been a few other reports of combined single molecule Fred and single channel recorders. None of them have been able to convincingly measure activity in Fred from the same molecule at the same time. So this is actually still the first where it was, you know, sort of convincingly shown. And they reiterate the point that Paul Selby made, that this type of measurement is necessary to relate conformational heterogeneity with various functional modalities that are typically obscured in ensemble measurements. And then a very important sentence, some of the technical difficulties associated with combining two single molecule techniques have been reviewed elsewhere. And this is a rather a large review, so there's a lot of technical difficulties. We tried to do this with actual ion channels like the KCSA channel, for instance, in later years, and while we were able to reconstitute these channels very nicely in GUV-derived membranes, we had a big problem with the optics because we had too much background fluorescence. So that is still a real big problem to actually achieve this. And the reason is partly that the technical difficulties of single molecule voltage clamp fluorometry are sort of multiplying each other, you know. So you have, the experiments are complex. You have to have a membrane that has to be stable. You have to reconstitute your channel. You have to have low background and so forth. And failure is likely at each step, and conditions for success are met transiently at best. So if you do a calculation of your success probability, each step has a success probability significantly lower than one, and the total success probability goes down very, very quickly. So what you need is probably, you need to do a lot of experiments simultaneously. So you need array formats. That was my reasoning. You need to do this experiment in a format where, okay, if one membrane breaks, then you go to the next membrane. And if one fluorophore bleaches, you go to the next membrane and so forth. And so the solution would be parallel array formats, but these parallel array formats have to preserve the high resolution, both optically and electrically, importantly, electrically. You know, many people have tried some similar stuff, but in situations where the electrical resolution was more or less lost because they had very large capacitances and stuff like that. And that's not good. So we thought that the microelectrode cavity array that we came up with in 2008 might be a good solution if the MECA, yeah, if you could make it so that it could be addressed optically. And so the microelectrode cavity array, I think most of you know it already. It's a device where you can make synthetic membranes on top of these little cavities, and you can insert channels or pores, and you can do lots of high resolution recordings. The resolution is really good because due to the arrangements where you have little microelectrodes here on the bottom of each well, very near the membrane, you have very low capacitance and you have very low noise. So the signal to noise is actually very, very good in this configuration. Now, if you can combine this configuration, which many people have used nowadays, this is just a little example of an experiment that you can do using polyethylene glycol and sizing polymers and stuff like that. So this is used a lot for nanopore experiments nowadays. If you can, and of course it's available commercially, I don't have to repeat this here, but you could see it again. So in two versions, one in 16 with 16 automated bilayers and one with 4 manual bilayers. If you can do this and preserve the good electrical resolution, but at the same time have optical access with a high numerical aperture objective to this membrane here, then maybe you can do something with this. And this is actually a very old idea that we came up with already in 2012. So this is a slide from a talk I gave in 2014 about this, where we had already started to make at least the structure. So what you do here is you instead of having a disc-shaped electrode, you have a circle, right, or a C-shaped electrode, which leaves an optical window through which you can actually see the membrane. This SU8 layer here is 20 microns, so you're near enough to be able to actually focus the membrane with a oil immersion objective, for instance, oil or water immersion objective with high numerical aperture. So this is the device, how it looks. This is one, I think this is an early version here of the MECA-opto or MECA-opto inverted, as it's called now, because there's another one where you can look from above, which doesn't have the high optical resolution. So here you have a C-shaped silver chloride electrode. Here you have the cavity, and over this cavity you put your lipid bilayer, and you can use an oil immersion objective to do this type of recording. This is a newer version. This is sitting on top of a microscope. You just have this extension here to the Orbit Mini, so you can use the Orbit Mini and the elements amplifier inside to do these experiments. And this is the basic layout. So here you have the electrode, here you have the SU8, here you have the glass, and here you have your membrane. So you can have a lot of fun with this to start with. You can label your lipid with fluorophores, and you can apply voltage, and you can see the bilayer breathe, rhythmically as you change the voltage due to electrostriction. So this here is the actual dye layer, which has very low fluorescence because there's only a bimolecular layer. Here you have your annulus, which is thick and therefore fluoresces a lot. You can also apply very high voltage and see how the membrane breaks. If you ever wonder what happens with your lipid when your membrane breaks, this is what happens to it. You can see the membrane float away, into oblivion, so to speak. But you can do other things. You can, for instance, put it on a confocal microscope. In this case, this is a microtime from PicoQuant, where you can actually look at lifetime. You can do lifetime imaging. This is a Z scan through this chip here. You can see here you have the glass, and you can also see one of the problems, which is that the glass has some autofluorescence, which has a short lifetime, but so we can easily distinguish it here. But it's there. And here you have the membrane. And the membrane is not really planar, as you can see here, but it sort of sags a little bit. Sometimes it sags in this direction. Sometimes it's the dome shape in the other direction. Depends a little bit on how you actually make your membrane. That's very important. So you can control it to some extent. Some people have learned to do it. Toby over there, for instance, he has some idea, what to do. If you want to have a membrane that is really very, very flat, which for some things is really, really important. So we've put it on a number of microscopes, also this one, for instance. This is the wonderful new Luminosa from PicoQuant. And you can do, lifetime measurements. For instance, here we have labeled lipid, lissamine, rhodomine, PE, and we can recover very nicely the lifetime from the literature. Here we have something more interesting, which is a labeled peptide, ceratotoxin, which we got first from, not the labeled one, but first from Michael Meyer's lab, which is really nice because it forms channels almost like alamethicine. Some of you may know alamethicine, which is one of these barrel stave mechanism peptides. And we also find the right lifetime here for ceratotoxin, and we can correlate that a little bit with where we can see at the same time, we can see the single channel activity of the ceratotoxin. Here we have the labeled ceratotoxin, and here we have the native ceratotoxin. This ceratotoxin is, as I said, it works a little bit like alamethicin. So it forms channels by reorienting in the membrane under voltage and then coming together to form these channels here. And we saw some interesting behavior of the labeled one. It seems that the C-terminal fluorophore, the atto, does seem to block the channel to some degree here, so that you can actually don't see really very well this opening. So there's an interesting interaction here. What we also saw is that the lipid associated peptide shows some polarization anisotropy that disappears when we actually break the membrane. So here we have a direct correlation between the photon count from an area of interest in the membrane and the opening and closing of the individual channels. Unfortunately, we do not see any signal because there's no reason for it. We just could actually see the fluorophore in the labeled peptide in the membrane and correlate and at the same time simultaneously do high resolution recording. And then if you break the membrane, or the membrane actually breaks spontaneously in this case here under the influence probably of the opening of the channel, then you can see that the polarization anisotropy disappears. So it means that there's a strong association between the fluorophore and the membrane actually. So what we also want to do is, of course, we don't want to do only film or lifetime measurements. We want to do widefield microscopy, possibly single molecule resolution. And for this, we teamed up with a group of Max Ulbrich in Freiburg, who are experts on this. And here is a little experiment that Frederick Görlitz in his lab did, just with a normal confocal microscope and a few beads. And these beads are just put on the surface of the chip, and you can see nicely here the beads on the surface in the bottom of the chip, and here the beads on the surface above. And this is important because this can give you information about the optical quality of the recording that you can get. Here you have the XY, and here you XZ and YZ extent of the fluorescence. And what you can see here that, of course, it's smeared out a little bit at the edges. Yeah, so there are edge artifacts here that you have to take into account when you use this device. Frederick came up with this cartoon here, who basically said, okay, the field of view that you actually get where you can get really good resolution is sort of in the middle, because the electrodes, of course, they do some shadowing. Yeah, you get some shadowing artifacts. So he said it's outside here, it's okay-ish. Here it's really bad, you know, above the electrodes, but in the middle it's really good. And our answer to this would be what we could do, and we probably will try to do that very soon, is to make a new version of the chip where we actually have the membrane smaller, the extent of the membrane smaller than the extent of the whole chamber, using a double layer design. That was actually the first design we came up with, but it's very difficult to make. Yeah, it's much more difficult to make. But in that case, you would have a field of view which is really optically of high quality. Okay, and finally, where we are now, we have now built a microscope that is really adapted to this. And with some tweaking, we are now able to actually see single molecules. So what you see here is single lipid molecules. Now we increase the laser density, so it's slowed down now. And you can actually see this is single molecule resolution that we have obtained. We haven't yet done this with labeled ion channels or labeled pores, but we are about to do that. And I'm very curious to see how far you have got in Lausanne with more or less the same idea in mind. Yeah, and that's basically what I have to say. And I thank very much, especially this guy here, Max Ulbrich in Freiburg, and also, of course, Frederick Görlitz, who did these single molecule recordings, and Tobias, who was instrumental in getting out the first paper on this. Okay, thank you very much.
Well, thank you so much, Jan, for this thorough and fundamental introduction. Excellent. Are there any questions from the audience?
Thank you very much. This is a great talk and great job. And I was wondering, what is the effective field of view? You mentioned that it's good in the middle, but you didn't mention how big it is.
Yeah, it depends, of course. It depends, of course, on the size of your chip, right? I mean, we have several versions. I mean, you have a 50 micron, 100 micron, 150 micron, I think even 200 micron chips, and then that would depend on, and it depends on the height of the SU8, right? I mean, we're also trying to maybe reduce the height of the SU8 because that would also increase your numerical aperture, actually,
Right.
because you have a sort of effective numerical aperture that you can calculate. I'm afraid I don't have it in my head. So, but it is of course possible. Yeah, I mean, Frederick Görlitz would know it. Yeah.
Okay.
Yeah.
I was also wondering whether the lipid composition influence the field of view or effective
The
area.
lipid composition, no, I don't think so. The lipid composition doesn't change so much the geometry of, or actually doesn't at all change the geometry of the whole thing. No, no.
Okay, okay. Thank you.
So just maybe a comment from my side. You pointed out that it's actually quite challenging to have a good optical readout and a good electrical readout. And I remember when I did my PhD with Claudia Steinem, we were working on conic scenes, and we labeled them with GFP to investigate them under a confocal microscope. But then when we did the electrical recordings, we found crazy subgating, which was probably the GFP wobbling around right in front of the aperture.
Yeah.
So it's actually quite challenging to get everything right.
Yeah, I mean the interference of the label with the function of the pore or the channel. This is something you could see in the ceratotoxin experiments, right? I mean the label was actually blocking the channel. I mean if we had a lot of time, we would maybe investigate that, because it's actually quite an interesting phenomenon. You know, it's like you have a little inactivation gate that adds to the channel. Yeah.